You can start whenever you want.
Okay, thank you. Thank you guys for your invitation. I'm really glad uh, to have been invited, also because this is a good, say, situation to share, I mean, discuss about every time about the results of every research, and also to speak with uh, people that I consider like friends in this field, like you, Maria, or James and Irina, that I met also at Bilbao. Um, so I start sharing my screen, and please, if you have any issue to visualize the presentation, let me know. Okay, so in principle, you should be able to see the presentation now, right?
Yeah.
Okay.
Yes.
Okay, great. So, as Maria said, I have been granted with this funding from the University of Padova in the north of Italy, where I'm actually working. And the focus of this research was to analyze the evolution of human microbiome, focusing especially on the Neolithic transition. So as probably, I think most of you already know what are we talking about when we refer to the Neolithic period. But basically, the Neolithic is that moment in our recent history where our species let's say started to be from started to passage from from an hunter-gathering lifestyle to an agricultural one. And this period had, let's say, several changes associated. It is the moment where we start to create, let's say, a more complex social organization, and also where when we domesticated plants and animals. And most importantly, we experienced also an important demographic increase during this phase. In Italy, the Neolithic transition came with, as for the rest of Europe, with the population migration from the Anatolian region, and the first Neolithic let's say sign that we have in our peninsula are referred to 6,200 years before Christ. In this, especially in the south of Italy, in that part that we call it today Apulia. The Neolithic was not a monolithic event. Indeed, depending on the geographic area that we are considering, this transition had, let's say, developed some specific features. So there are, for example, situations where the transition was not so strong from, and so we cannot see very, let's say, stronger signal of transition from the Mesolithic period to the early farmers community, and other situation where instead there is a big shift from an archaeological perspective from the Mesolithic, from the last hunter-gatherers societies to the early farmers individuals. From a microbiome perspective, as you already know, we had some controversial results, since from the very first study that focused on the ancient dental calculus microbiome, it was suspected to have a shift in the microbial composition of dental calculus during the Neolithic period, while in more recent research, such as the one of Ottoni or Fellow, they were not able to replicate this result and to detect, let's say, signals or relevant signals of transition in microbiome composition related to the Neolithic transition. In the study of Ottoni, they found only 2 bacterial species that could be, that, let's say, associated with this transition. That was, if I remember correctly, Olsenella or Altaxon, and one Anerolineaceae species. So the aim of this study was basically trying to understand if there was any ecological shift experienced by the human microbiota that could be associated with the agricultural transition in the Italian population, and also trying to understand how these new subsistence strategies could have affected the microbiome composition. To find an answer to these 2 questions, we wanted to, let's say, try to improve some of the aspect in the field related, for example, to the sample collection. We know that from what we know about most of microbiome research, environment is one of the main, let's say, variable that it's able to influence the microbial composition together with diet. Although for the oral microbiome this is still questioned, we can say, but generally speaking, the environment is one of the most known forces that shape microbial composition. So we noticed that in most of the ancient microbiome literature, sample that were compared came from very different environmental condition and geographical region. So we wanted to try to reduce the possible effect of environmental variables, collecting samples that belong to a very specific area, which is the central south of Italy, where, as I said before, the Neolithic transition started. And we collected sample that came from one last hunter-gatherer population that was always in the same area from the Paleolithic period, and also several Neolithic sites, which are the one in oranges, and also some post-Neolithic sites, which are the yellow one, which referred to the Neolithic period. Then something that we want to improve was also to deeply integrate the ancient DNA analysis with the archaeological information and also with the anthropological analysis, and also performing the detection of vegetal microdebris, always from the same sample from which we collected the DNA. Moreover, one last part that we wanted to improve was to consider all the archaeological phases of the Neolithic, at least in this area. So we have here 4 main archaeological phases, which are called the early Neolithic, middle Neolithic, final middle Neolithic, and late Neolithic. So we collected samples that belong to different sites corresponding to each one of these different cultural background, which was also missing in the previous literature, since normally the Neolithic sample were just considered as Neolithic, but without paying attention to this to their archaeological or cultural background. So first of all, we try to we create a network basing on the microbiome composition among all the sample, and we wanted to detect if there was any significant cluster among this network composition. And we found, let's say, more cluster than expected in truth, because we found 5 different clusters. So after, we wanted to understand which one of our variable, let's say available variable from archaeological or anthropological variable, could explain better this cluster distribution. And we find out that the period, along with the site also, for site I mean the geographical location, were the best feature to describe this cluster distribution. So then we went to associate let's say the cluster subdivision with the period. And we observed that, for example, cluster 1 was composed almost exclusively from the Paleolithic samples, so from the hunter-gatherers individuals, while the second cluster was characterized mainly by Neolithic sample, but as you can see here, it was composed mainly from sample that belong to the first phases of the Neolithic period, so from the early and the middle Neolithic. The third group was instead composed mainly by sample that belonged to the last part of the Neolithic transition, so to the late Neolithic, and also to the Copper Age, or Neolithic as I called it before. At the same time, the fourth and the fifth clusters were, as let's say, were composed by sample that belong to the last part of the Neolithic as well, but also, let's say, these last two clusters were composed by sample that belong to this phase, to the end of the Middle Neolithic. This was something that, let's say, we pay attention to, because the final Middle Neolithic sample that belonged to the final Middle Neolithic came from this site, which is called Palagiano. And so the sample that belonged to this same site were split in two. And this was, let's say, almost the unique situation that we found among our data set, where people that come from the same site were divided into different groups. And so comparing our information with the archaeologists, we found an interesting trait that for the moment is just an hypothesis. The archaeologists noticed that in this site that there were two different burial how can I say, burial traditions that were living together in the same site. And the most individual that came from one of these two clusters came also from a same burial tradition, we can say, while the other no. So this was just an hypothesis that there could be a somehow correspondence between these two information, but we don't have for the moment any more clues about. Probably performing a genetic analysis, we can have more clues about this information. Anyway, we compared the microbial composition in our from one cluster to another. and we detected that there were some important statistical difference in the microbial abundances. So we didn't find any change in presence or absence of species from one period to another, but we found a difference in the in the proportion of every of these species across time. Principally, we found two trends. One, which is an increasing trend of species that were poorly present in the hunter-gatherer samples and start to increase their abundance with the beginning of Neolithic, and it's only at the end of the Neolithic period that they definitely increased their relative abundance. And to this group belong species of the red complex or orange complexes, as you can see here. So there are species which are very poorly present in the Paleolithic period, and with the starting of the Neolithic period, we have an increase in their abundance, but it's only at the end of this phase that we saw an increasing in their relative abundance.
Andrea,
See?
sorry
Yes.
if I interrupt you.
Mhm.
Could you move this pop-up window? Because I don't know if you see it, but we all see a pop-up window at the bottom saying that Teams
Sorry.
Yes, maybe if you
Sorry.
can move it or not.
Yes.
Well, perfect. Thank you so
I'm
much.
sorry.
Sorry for interrupting.
No, don't worry. And but a part of the species that belong to this well-known complex for the oral microbiome, there are also species which are part of the, let's say, normal oral microbiome that follow the same trend. One, above all, was the Solzenella oral taxon, that, as you can see, I mean, it was poorly present in the Paleolithic sample, and only with this beginning of Neolithic, it increased enormously its abundance. And this is the same species that was also observed by Ottoni in this study. There are also species that I said before following let's say the opposite trend, so the decreasing trend. These are species that are mainly present in the Paleolithic period, and they start to decrease their abundance with the beginning of the Neolithic. And at the end of the Neolithic period, they are very poorly present. Sorry for I changed the desktop, and then now you cannot see the title, but here it is written purple complex. So as you can see, there are species that are following the opposite trend of the one that I described before. At the same time, we focused also on the functional pathways of these microbiomes, and always comparing these functional pathways between among this cluster that we detected, we found some significant differences in several pathways. For example, as you can see here in the carbohydrate pathways, there are, as we've seen before for the species, pathways which are particularly enriched in the hunter-gatherers group and which tend to decrease with the beginning of Neolithic, or pathway that follow the opposite trend, as you can see, for example, for the galactose metabolism, or also for the glycan pathways. Something that was interesting to observe is the correspondence between the microbiome data with the paleobotanical information. Indeed, as I said to you at the beginning, from the same individual, we performed also microscopic analysis and identification of the microdebris. And for example, we found that Paleolithic samples were particularly enriched in microbes involved in starch and sucrose metabolism with respect to the following period. And also that the hunter-gatherer sample were all particularly enriched in, let's say, a huge starch grain morphotypes than the Neolithic sample. One interesting point was that we found also in the dental calculus of this Paleolithic sample some, let's say, traces of avena, which was founded also on a grinder stone in this same archaeological site, and now the paleobotanist detected it also in the dental calculus, so confirming that this plant was consumed by the population. While in the Neolithic sample we have a strong reduction in the morphotype, which are mainly the grains and legumes which are part of the Neolithic packaging. So probably we suspect that these changes in the carbohydrate pathways could be a sort of a reflection of changes in the vegetable source, in carbohydrate sources from diet. At the same time, we found also differences in the amino acid pathways. And we tried to find if there was any possible correspondence from what we were able to observe from the carbohydrate or amino acid pathways in the Neolithic sample with what was recorded in the scientific literature about the oral composition, about the oral microbiome, since we have few data from dental calculus association with diet in modern population. And we found that some of the let's say, functional pathways that mostly characterize the Neolithic samples, such as the cysteine or lyseine or arginine metabolism, were also found to be enriched in nowadays vegetarian population, which let us hypothesize that with that this Neolithic population could probably have shifted their diet to a more, let's say, vegetarian diet with a lowest intake of meat, and also with a reduction in the biodiversity of this plant. And this hypothesis could find possibly a confirmation, let's say, from the isotopic analysis that was conducted from other researchers on sample that belong to this same geographic area, where they observed that from the early Neolithic to the late Neolithic and on the following period, there was a reduction in the meat intake in their diet and an increase in vegetable intake. So their diet became, let's say, reduced in the vegetable biodiversity, and mainly relied only on vegetable resources. One last interesting point was that with the increase of the red and orange complexes, we found also a correspondence with an increase at the end of the Neolithic period of the virulence factors. And comparing this information with the anthropological data that we have for every population that we sampled, we found that anthropologists recorded a low presence of periodontal disease in Paleolithic and early and middle Neolithic sample, while a strong increase in the oral pathologies among the late and Copper Age samples. Finally, we were able to reconstruct the genome of this Olsenella oral taxon, which is let's say an interesting species since it is present. It was also recorded in the Ottoni study, as I said before, to be associated with the Neolithic sample, as we also observed in our data set. And also because if we compare the Neolithic sample from the Italian region with the sample from the rest of the world, we saw that this species was very present only in the Italian samples. So we want to understand how these species have evolved during time. And we found some interesting aspect if we compare all the ancient strain that we reconstructed, that were 6, with the modern reference. And particularly, we found that there are some regions that are missed in the ancient, let's say, genomes, that were related mainly to defense mechanism and interaction with the rest, I mean, with the other species of the with the other species. Since the bacterial interaction is one of the main forces that can shape the microbiome composition, we can also suspect that probably the evolution of the single species can have an influence in the evolution of the global microbiome. So, going back to our question, if it was possible to detect any possible ecological shift experienced by the human oral microbiota during this period, we detect, we can say, 2 main shifts, at least in our data set. One related to the introduction of agriculture, and but also a second shift which distinguished the first farmers to the second part of the Neolithic. And about how these new subsistence strategies could have affected the microbiome, we hypothesized that these changes in the bacteria species that we observed could be probably related to an increase let's say in the agricultural practice, but generally speaking also in the dietary resources that this population selected over time, and that this change in diet could probably have selected some of these species, such as, for example, the pathogenic oral bacteria, with also, let's say, a bad effect on this population health condition. So I thank you for your attention, and of course I thank you all the people that have collaborated to this project, which are the one listed here.
Thank you, Andrea. So anyone, if you have any question, please feel free to post both in the chat or, yes, as Miriam said, to unmute yourself and just ask any questions. We have 25 minutes, so feel free to go wild.
Yeah, I think Irina has a question.
Can you hear me?
Yes.
Okay, great. Yes, thank you for the presentation. This is really interesting.
Irina.
I have a question about the glycan results that you showed. A couple of those are surface markers. LPS is specific for gram negatives, and peptidoglycan is much more abundant on gram positives. So did you look at all to see whether or not there's a transition in the abundance of gram negative and gram positive species in your communities through time?
No, I didn't observe if there was a, let's say, this macro change about a gram negative or positive on, I mean, in the overall. I mostly look at the single species, I must confess. But it's an interesting point that we can maybe think about to do.
Yeah, it'd be interesting because there's some debate in oral microbiology research today that suggests there's more gram negative species associated with disease and more gram positive associated with health, but that's not really strongly shown. So it'd be interesting to see if there's a shift through time.
Yes, thank you for this suggestion.
You can ask on your side.
Yes, thank you for a great presentation. I have a question about genome assembly because you presented Olsenella, but I have a question. Did you find any other high quality or medium quality de novo assembled genomes in your data set?
Yes, thank you, Piotr, for your question. I hope that I pronounced correctly your name, sorry in case. Yes, in fruit, we found also other species that could be assembled that had a good quality. We decided to focus it on the Olsenella because for two reasons. The first one is because, as I said before, it was strongly related to the Neolithic. And Olsenella is something that, as said, it was, let's say, very abundant in the Italian samples, but not in the other samples, I mean, from that were analyzed in other studies. And so, I mean, I was very curious to know something more about this species, also because Olsenella is not so common in the modern dental calculus. I worked also on modern dental calculus sample, always here from Italy. And yes, you can detect something of Olsenella, but not so much as it was observed in this in this Neolithic sample. So I wanted to know why, what happened to this species, why it was so abundant only in the Neolithic, and why today is not the same situation. So probably there are other shifts, I don't know for the moment. And the second reason was because it was transversal. So we were able to reconstruct Olsenella from sample that belong to the Middle Neolithic, Final Neolithic, and Copper Age. So they were not only from one unique phase. So this is why we focused mainly on Olsenella, but in truth, we had also other species that could be reconstructed from the data set, and we are currently also working on this.
Okay, thank you.
You're welcome.
Yes, I have a question on there.
Yes.
Yes, I think that it was very interesting, that thing that you mentioned about Pelagiano, the two burial traditions. Could you tell a little bit more? Do you refer like having individuals with different, maybe social status or something related?
Yes. Yes, thank you, Miriam, for this question. Yes, I presented you that case because it was a very particular situation. and because I thought that could be of inspiration for other people to look at the microbiome data also in this sense, also from this perspective. But I must confess that in my case, I don't have so many information more, because this site called Palagiano was not so extensively studied from other perspectives. So we only know that there we have different burials, and that at least archaeologists recorded two burial tradition. And what was interesting to observe was that one of the two clusters was composed by people that belonged to a same tradition, to a same type of burials, while the other, no, there are individuals with different tradition in the other cluster. So archaeologists suspect, basing on the archaeological data, that there was social differences in this site. And the fact to find some differences also from the microbial perspective is, I think it's very interesting. But for the moment, I cannot claim so surely that there is not a perfect correspondence, of course, between the burial tradition and the microbial composition. But the fact that we found these two clusters, and there are also this, let's say, social aspect, can maybe give the idea, or this, the hypothesis that probably we can use microbial data also to, I don't know, maybe inspect this lifestyle aspect. But for what we know, actually, I think from dental calculus microbiome, it's very hard to find to answer in a clear way. We know still, very poor information about. But maybe what we were planning to do was also to perform a genetic analysis on this individual, to see if, for example, this burial tradition came from a different host genetic background. So maybe like two populations that migrated together, for example, in that site, that lived together in the same moment. So maybe that they had, I don't know, maybe also different dietary habits. So we are planning to perform also the isotopic data on that. So for the moment, unfortunately, I don't have an answer, but I hope to give you an answer soon.
Thank you. Thank you.
You're welcome. Yes, I see the question of Aida. Yes, there are some data that on the isotopic profile, but they were related to the strontium, so which are mainly associated to possible migration phenomena. So for the moment, we don't have, let's say, the dietary isotopic profile, but yes, this is one of the, let's say, project that we are actually performing. Were they all local? That's a good question. As I said, until I don't have the genetic background of the host, so of the human, I cannot say, all the genetic story, but from the strontium level, they seems to be, let's say, of that geographic area, not specific, let's say, not specifically local, but from that background. You're welcome, Aida.
Okay, I guess I also have a question. So before you mentioned that you saw some virulence factor, like pathways for virulence factors increasing, and did you see only this increase towards periodontal disease, or did you could you see patterns for other disease other than periodontal?
You mean like on the bones, if this skeleton remain have also other
Just from your analysis, was it specific for periodontal diseases?
Okay, let's say unfortunately yes, because the anthropological data that I had in common for I had different site as I told you, and each one of these sites was analyzed by different anthropologists over time. So and these data were published. So not in all publication the anthropologists recorded always let's say pathologies on this on all the body or and also the oral. So there are some studies where I had only the oral pathologies and other where I had also let's say pathological marker from other part of the skeletal. But I mean, I found like a minimum common denominator between the oral pathology. So I only used the oral pathologies that I had as information. Also because I thought that they were more direct to correlate with my microbial information. I mean, I was not so sure to be let's say robust enough to correlate the my data with possible pathologies in the rest of the skeletal. I don't know if that could have any biological, let's say, significance, so I only concentrate on the oral.
And could you see the actual sign of the periodontal disease on the fossils, or there were cases where you would see the pattern but not the sign on the tooth?
In truth, no. I mean, it was not point by point. I mean, I did not perform a correlation between the presence of virulence factor and, for example, the presence of the number of caries. Also because I think there is not for sure a so direct connection, in my opinion. What I did was to observe for every site and for every period where I had the most I mean, the significant increase in the virulence factor, and then I go back to the anthropological data to see how the anthropologists were describing that sites. And I saw this interesting correspondence, because in the case where I found an increase in the virulence factor, in their publication, the anthropologists described that that site as being strongly affected by different type of periodontal disease that could be caries, huge dental calculus amount, loss of teeth, while where I have a very low presence of this pathological, let's say, bacteria, the anthropologists were describing a situation with a very low amount of this pathological condition. So it is just, let's say, an association between their description and what was I observing.
Okay, thanks.
Andrea, I have another question.
Yes.
It's like a more general one. Could you share with us just one or two, or one of the major challenges that you face doing this study? I think that behind every paper has like challenges, so it will be great
Yes.
if you could share with us.
Well, the most important challenge at the beginning, it was, as for everyone, the COVID situation, because my research started with the beginning of the COVID pandemic. If you remember, in Europe, Italy was the first nation to be hit by this pandemic. So we were among the first to go in a lockdown situation, exactly when I was performing the collection of samples. So it was not a good moment for the lockdown for me. And this, I mean, under a practical point of view, we really need to do an effort all together, all the people that I presented you in the last slide, because there was I mean, we had to perform a sample activity all together to be able to do that in time as it was scheduled. So that was like the big problem at the beginning, was that one. And the second was to find the sample condition that I was that I wanted to find. So trying to reduce this environmental variable, so trying to get sample that belong to only a specific area, and that go from a very long temporal range, so from the Paleolithic to the Copper Age, always stay maintaining this same area, having a good sample size, so having at least enough sample to describe one site, every site, and also to have also to have the archaeological and anthropological information for every of these sites. So, I mean, the inclusion criteria were very difficult to let's say, to respect at the beginning, because most part of the beginning of the work was exactly to select what was for me the perfect condition to perform the analysis.
Thank you. Thank you for sharing.
You're welcome.
And I think, connected to this last comment that you did, you mentioned in the beginning that you tried to reduce as much as possible, like environmental variables.
Geographical variable, let's say.
Geographical variable. But do you think that all the changes that you are seeing, they are really like only taking into account just the diet variable?
Of course, I cannot be 100% sure, because it's something that it's something that we are observing from our past. So basing on my information, on what I know about the lifestyle of this population, and basing also about what I know about the influence of diet on microbiome, I think it is more easy to suspect that, let's say, the main actor can be diet and let's say dietary choice also. But as I said in the last part about Olsenella, I suspect in a very strong way that also the influence of the single species evolution can affect, as a consequence, the composition. So all these species are all species evolving together, one to another, and they are sharing their genetic material. So I think that one important also force in shaping the microbiome, but it's not just, it's not like my hypothesis, is something that other people is describing, is the genetic evolution of species that can influence also the relation between species, and so also the composition of this microbiome. So probably I suspect that there are multiple factors that can explain the situation that I'm observing. So probably I don't know, also the fact that another population can come having their own microbial composition, and so maybe affecting a little of this overall microbial picture, the evolution of the single species, diet, of course. So probably there are multiple aspects that participate in creating these observing changes. But I suspect that diet, at least as I repeat, based on the information that I have to date, was the most evident, let's say, correlation with what was I observing. Also because I had isotopic data from the same population and also other population that came from the same area and the same period. And from the isotopic data, they observed the difference in the diet and in the dietary choice between the beginning and the end of the Neolithic period in this area, of course, because probably in other area is not so say that they had the same situation. There were also an interesting paleobotanic study that was performed always in the south of Italy that recorded in the last part of the Neolithic period a rapid climatic change with an increase a strong increase in the main temperature. And from the paleobotanical remain, they observed that this population started to select more resistant plants to that specific, let's say, hot temperature. So, I mean, we have indication that this population changed something about their diet, maybe for cultural choice, maybe for environmental choice, but also from the isotopic data, there are information that reveal this change in their dietary choices. So for this reason, the connection with what was I observing on microbiome data was more immediate, at least.
Thank you.
You're welcome.
Any other last minute questions? Because very soon I think we close. So if you have any last quick question for Andrea. Okay, so it looks like we can conclude. So I'm going to stop the recording and